Hello, in this video, we'll be seeing about enzymes. Proteins. Proteins are polymers made of monomers called amino acids. All proteins are made of 20 different amino acids linked in different orders. Proteins are used to build cells that act as hormones and enzymes and do much of the work in a cell. Enzymes. Most enzymes are proteins that are tertiary quaternary in structures. These enzymes act as a catalyst to accelerate a reaction. They do not permanently change the process and are specific of what they will catalyze. Enzymes are reusable. Their names usually end with ase, such as sucrase, lactase, maltase, etc. Enzymes are natural catalysts. Up to 10 to the power 16 over uncatalyzed rates can be gained. Only the desired reaction which you want will occur using enzymes. Primitive reactions can occur under mild conditions. Since most of the reaction in your body's cell needs special enzymes, each cell contains thousands of different enzymes. These enzymes let chemical reactions in the body happen millions of times faster than without the enzyme. Because enzymes are not part of the product, they can be reused again and again. One example of enzyme is restriction enzymes. These are recognizes specific base sequences in double helical DNA and cleave at specific places both strands of the duplex containing the recognized sequence. Restriction enzymes recognize the specific base to pair the sequence in DNA that is called the restriction site and cleave the DNA by hydrolyzing the phosphodiester bond. The cut occurs between the third carbon of the first nucleotide and the phosphate of the next nucleotide. The restriction fragment end have 5 phosphates and 3 hydroxyls. Most restriction enzymes occur naturally in bacteria. They protect bacteria against viruses by cutting up the viral DNA. Bacteria protects their DNA by modifying possible restriction sites, such as methylation. More than 400 restriction enzymes have been isolated. Names typically begin with three italicized letters, such as Ecorel or Hind III or BAM HI. Most restriction sites are palindromes of 4, 6, or 8 base pairs, and the short restriction site sequences occur more frequently in genome than long restriction site sequences. For example, 1 by 4n. The application of recombinant DNA technologies. First is pharmaceutical products. Insulin. Cheaper and safer compared to the animal insulin can be created. Vaccine subunits against hepatitis B is safer since it will not be infected by pathogens. DNA of vaccines against malaria, influenza, etc. can be gained. Gene therapy. Replacing defective or missing gene with normal gene using adeno and retrovirus or vector. Gene silencing, known as RNA interface, using dsRNA called short interfacing RNA that targets specific gene and degrades it. Thank you. In the next video, we'll be seeing about hormones.